Hello everyone, welcome to BioCaryon. Today we will discuss the topic of DNA replication, first of all in prokaryotes, and in the second video we will discuss about the eukaryotes. See, DNA replication in prokaryotes. See, the prokaryotes have a closed circular DNA and have only a single chromosome. Prokaryotes means I am talking about bacteria, or you can say specifically E. coli DNA replication. Now they have a single chromosome, and this single chromosome is also circular in nature, not like the eukaryotic chromosomes which are linear in nature. Now this single chromosome is like this. I mean to say it is double-stranded, right? So it is a double-stranded chromosome, and somewhere it has a point, it has a position which is known as ORI-C. ORI means origin. This ORI-C is the origin of replication in case of prokaryotes. This ORI-C location has certain base pairs which are 13 base pairs in length. These are 13 base pairs in length, and it has a stretch of repeats of these 13 base pairs. These 13 base pairs, suppose these 13 base pairs are present in this region. These are known as, the 13 base pair region is known as 13 mers, because these are of 13 base pairs. Ahead of it a point is present which is known as 9 mers, and these 9 mers have 9 base pair repeats. So first of all, in DNA replication in case of bacteria and in prokaryotes, the position this 9 mers, suppose I make this position like this. This is the 9 mers position, this place, this region. Suppose I draw it like this. So what happens? Initially, this 9 mers, they supercoil among themselves. They coil over themselves. How do they coil? They coil over themselves. So this is the region of the 9 mers. And a protein that is known as DNA A protein, it makes this 9 mer region to bind over itself. This protein is known as DNA A protein, makes the 9 mers to bind over itself. So when these 9 mers, they bind over themselves, if I change the diagram slightly, after binding over itself, this 9 mer region comes to be located in this position. So if it was, it will become like this, right? With DNA A protein that I have drawn over here coming in between. Now when 9 mers supercoil over themselves and DNA A protein comes to bind over here, then they are attached. You can see 13 mers is present over here, which is actually the origin of replication. At this time, what happens? There is an enzyme which is known as DNA B. This enzyme that I said was DNA A. This one is DNA A. So another enzyme that is known as DNA B, which is circular in nature, just like this. This is circular in nature. This enzyme comes. Now you can see a small clamp. This is like a rope, and inside the rope you are putting this clamp. This is a ring-like structure. This is a ring-like enzyme, and it comes and attaches to this ORI-C position. Now this ring-like structure is actually a helicase. This is a helicase enzyme. What is helicase? Helix is. Ase means breakdown. Ase means breakdown. So this helicase enzyme, what it does, it breaks down the helix present at this position, right? And this helicase enzyme is not alone. It comes along with another protein, that is DNA C. DNA C is a helicase loader. I will draw it over here. This is DNA C. Now you can see 1, 2, 3, 4, 5, 6 subunits are present in DNA B. Similarly, if you count them, 6 subunits are present in DNA C. Now when DNA C comes to this position of 13 Ms, it is released, and DNA B in circular fashion, it's just like a ring, it comes and binds to this 13 Ms position. Right? Now how it binds? If I pick up a strand, if this was the DNA strand, so if this was the DNA strand, it will open like this, right? And the helicase enzyme, which I had drawn over here, it will come and attach to this position, and this blue portion, that is DNA C, will be released, and the red portion will be attached to this side of the replication unit. Now then, what is this? This replication unit is known as replicon, or you can see it looks like a bubble. It is also known as a replication bubble. This is a replication bubble. Now this is an unbinded DNA. It is not a double-stranded DNA. It has sites for attachment of other proteins, right? So if you don't protect these sites for attachment, DNA can be damaged or it can re-anneal to itself. So at this time, certain proteins come and bind to this, and these proteins, these are known as SSB proteins, single strand binding proteins. SSB is single strand binding proteins, this protect. Now what happens? This helicase, it moves in a swirling fashion over here. In a fashion, it moves over here, and after some time it will reach this end. From here, from this part, it will come and reach the other end. One DNA B enzyme is enough. Why one is enough? Because if one DNA B enzyme, there are base pairs between them, hydrogen bonds between the two strands. If hydrogen bonds in one strand is open, automatically the second strand is also open. So there is a confusion that helicase comes and binds to both the sides. No, helicase, only one helicase is there, comes and binds to this position, and through a swirling fashion it moves to the other end, breaking the hydrogen bonds between the two and creating a replicon, replication unit, also known as replication bubble. Now this is the site where DNA replication will occur in prokaryotes, and in cell where it occurs in cytoplasm. Right? Please remember, in E. coli, every 20 minutes DNA replication takes place. So this process of DNA replication will occur after every 20 minutes. Within every 20 minutes, one time replication is going to occur. Now how the process continues? We have discussed two enzymes basically. One is DNA B, which is the helicase, and one is DNA C, which is the helicase loader, which helps the helicase to bind to this place. Now this is the location which I had drawn over here where DNA replication is going to take place. See, one important point to understand is DNA replication occurs in 5' to 3' direction. Right? So when this one unit, one replicon is opened up, one replication unit is opened up, then another enzyme which is known as primase. Another enzyme which is known as primase, it is uploaded at this end. This end is known as replication fork because it resembles just like a fork. So at the end, at this position of the fork, the enzyme primase is going to attach. Because it is attaching to DNA, it is also known as DNA primase. Function of this enzyme is to add up an RNA primer. Add up a RNA primer. Now without RNA primer, DNA replication will not occur because DNA polymerase the enzyme, next enzyme that we are going to load, that is DNA polymerase. You can see the name DNA polymerase. This enzyme carries out the process of DNA polymerization. So this DNA polymerase enzyme, this cannot initiate the process of DNA replication. It can only elongate the DNA, but it will not start the DNA replication. To start the DNA replication, this enzyme is required, which is more properly known as DNA primase, which adds up a RNA primer. Now, to one end, suppose this is the primer that is added. RNA primer, please note that always added at the replication fork region, right? And RNA primer, I am saying that DNA replication occurs in 5' to 3' direction, means this was the 5' end and this was the 3' end if replication was occurring in this direction. Means this was the 3' end of DNA and this is the 5' end of DNA. Alternatively, this is the 3' and this is the 5' end of the parent DNA, right? And RNA primase attaches, DNA primase attaches at the replication fork at 3' end of the template. This is the template DNA and this is also the template DNA, right? Comes and attaches over here, right? Now what happens in next step? This is one strand of DNA, right? And both the strands of DNA have to be replicated. I told you that DNA primase comes and attaches at the site of replication fork. So over here also RNA primase is going to attach. This is 5' and this is the 3' direction. Clear? Now we'll change the diagram a little bit. In the next step, what happens? Now I draw only this replication bubble portion. Let it be like this. This is the position, right? Arrow, and here your helicase is present. And one primer has been attached over here. This is 5' to 3' direction, and one is attached over here. You can make an arrow to indicate the position of DNA synthesis. After attachment to this 3' end, a beta clamp is attached. Now what is beta clamp? Beta clamp is this shape. Remember the hooks present in your bones. It is a hook-shaped protein, right? And it has points for attachment of DNA polymerase on one end. On its one end, DNA polymerase can attach, and another end. Actually, the sides are same. To this hook itself, this is just the same side, and to this side, it can attach this beta clamp. We are talking about the beta clamp. This is the beta clamp. This has certain hook-like positions on it. To this hook, clamp loader is present, and this side is polymerase 3. And which polymerase? It is DNA polymerase 3 enzyme. This can attach. So at the 3' end, this enzyme, this beta clamp, is going to come and attach with the help of this clamp loader. Clamp loader is again an enzyme which makes this beta clamp to come and attach to this side. Right? After coming and attaching to this side, see, binding site for DNA polymerase and binding site for DNA clamp loader are the same. Right? So after attaching this red one, this beta clamp to this position, clamp loader will be released. Right? So just a protein, it moves in the forward direction, it will move in this direction, and it will keep this DNA polymerase in this position over here. DNA polymerase should be present over here in this position. Now if DNA polymerase is this, so I should draw it like this. So this is the clamp. This is the clamp, and clamp loader is now released when clamps come to this position. Now you can see this was 5' to 3' direction, and this round thing, this blue one, this is the DNA polymerase 3. So function of DNA polymerase 3 is to add, what is the function? Is to add, is to polymerize the DNA, is to add new nucleotides to this thing, and these nucleotides will be complementary to this strand, right? So DNA polymerase will start attaching DNA in this direction, right? And similarly over here also the same thing happens. The clamp loader will be there, and then clamp loader will be released. Beta clamp comes over here, and DNA polymerase comes over here, and this DNA polymerase, clamp loader in this case, beta clamp will attach DNA polymerase over here, right? Will attach this way, and over here it will keep on attaching. Now if you notice, this DNA polymerase is like a ring. It is rotating in this position. It is rotating over here, right? So in rotating fashion, it is adding the nucleotides, and similarly DNA polymerase moving further and added nucleotides to this side. So this is DNA chain elongation, right? And initially that I discussed, that ORI-C and 13 MERS and 9 MERS presentation, that was the initiation of DNA replication. DNA replication divided into 3 parts. One is initiation, second is elongation, and one more part is there, that is termination, which we will discuss later. So I hope it's clear from here. Now next step, what happens? See, helicase I have drawn. This red colored helicase is present over here. This helicase will further unwind. This DNA is present in circular fashion over here, and like this. So it will further open this DNA strand. Helicase will move further, and again will leave certain strands over here, and here also this is present. If you can see, the bubble has enlarged, right? Now one of this strand, see, if you see it a bit carefully, this part has been constructed, and this red one is the RNA primer. RNA primer is present over here, right? Similarly, RNA primer is present over here, and here also the DNA has been constructed till here. Now this much portion again unbounded by the helicase. This strand is the 3' end. If you see here carefully, this was 5', this was 3' end. So this is the 3' end, this is the 5' end. So DNA synthesis will continue in this direction, the position that has been opened by the helicase, right? Over here, this is the 3' end, and this was the 5' end. So DNA replication cannot continue in this direction, because here unbound DNA is not present anymore. So what happens, after coming to this side, after coming to this position, what happens, this beta clamp, later on this beta clamp, if we draw it again, this through synthesis, this beta clamp has reached this position, and DNA polymerase has also come to this place. So this DNA polymerase will be released from here, and another RNA primer will be formed. One more RNA primer will be formed over here in 5' to 3' direction, and a second beta clamp, this beta clamp, a second clamp will be loaded. So again a second beta clamp will be loaded with the help of a clamp loader, and this clamp loader is further going to attach this DNA polymerase 3 over here, so that, and move in this direction until it reaches this position, so that further I will draw this part, DNA synthesis occur. Now this is the elongation part of DNA replication. If you notice it, some 3-4 things which I would like you people to know over here. First thing, this is your double-stranded DNA, the entire circular DNA that we were talking about. this resembles theta structure. So this mode of replication is also known as theta. This structure is known as theta structure, first of all. Second thing, it is also known as rolling circle replication because it is rolling. Further again, see, helicase is present over here. This round red color, so it will again open and it will further extend, and further synthesis is going to occur. Here DNA synthesis is occurring in this direction. If I draw it like this, DNA synthesis is occurring in this direction over here, right? Now check this. See, here only one RNA primer had been drawn. We had drawn only one RNA primer, and in this same RNA primer the synthesis is continuous. So this type of replication is known as continuous DNA replication, where only one RNA primer is required, right? And in this continuous replication, this strand comes to be known as leading strand, right? Because the replication is continuous. In the second strand, DNA replication had been halted at this position and restarted at this position. So this is known as discontinuous DNA replication, right? And this strand is known as lagging strand. This strand is known as lagging strand, unlike this strand which is known as the flaking strand. I hope it is clear. This is the elongation part. Now replication will keep on continuing in this direction slowly, and one more diagram I will draw after this until it reaches the other end. If you further draw it, so this replication bubble will further enlarge further, shortening this DNA, this like this, and this replication bubble now stop increasing, and this is the second strand that is growing over here. This is the second strand, and this first strand also increased in length, and didn't show this part, but actually RNA primer still attached. Now one RNA primer over here and second one over here, and maybe some third one over here. These red ones are the RNA primers, and here the replication is continuing in the leading strand in this way, and here replication is continuing in this strand in this direction. This is the exact process of DNA replication in prokaryotes. It's not finished. Now slowly it reaches the other end. We will talk about the third part of DNA replication, that is termination, in a separate video. Here we are discussing only the initiation and the elongation. Now after the elongation is complete, you need to remove these red things, that is the RNA primer. So RNA primer is removed with the help of an enzyme that is known as RNase H. See, RNase H is an enzyme. RNase H is an enzyme. RNase, it is a RNase, removes the RNA, right? So it will be able to remove these RNA strands from these positions and from leading as well as lagging strand, right? But in the end, when it reaches this point, I am drawing it like this, where it meets DNA and RNA hybrid. This is DNA hybrid. So last one will be, like suppose this is the RNA primer, this is the RNA, and this thing is DNA. So when it reaches the end, now what happens, RNase H comes at this point and it starts removing the RNA from this position, right? This was 3 prime, this was 5 prime, and this was 5 prime again, and this is the 3 prime position, right? So RNase will come and start removing this RNA except this last one. The last nucleotide RNase will not be able to remove. Last nucleotide is left. Now this last nucleotide does not remain like this. It looks like a flap. right? And DNA polymerase, this looks like a flap, right? And DNA polymerase 1, there is one more enzyme, that is DNA polymerase 1. This has exonuclease activity, right? What does DNA polymerase 1 do? It has exonuclease activity also, and it has polymerase activity also, right? So DNA polymerase 1, it starts removing the strands. In fact, it's not RNAs, it is DNA polymerase 1 which removes in prokaryotes. If this was the strand, it was DNA polymerase 1 which starts removing the strands from here, except the last one, except the last that it has left, and in the meanwhile it polymerizes the strand also. So it polymerizes the strand till here at this time, and in 5' to 3' direction, and exonuclease activity is 3' to 5' direction, right? Leaves a flap over here. So there is an enzyme, flap endonuclease, FEN, flap endonuclease. This removes this flap finally from this position, and is also removed, leaving a small nick. This nick is there. This is sealed by DNA ligase, right? So in this way, from DNA, this again we remove this RNA, it is again becomes DNA. So this entire red colored things are finally removed and resealed with the help of this DNA polymerase 1 enzyme, flap endonuclease, and DNA ligase. So this is about initiation and elongation in prokaryotes, and in our next video I will discuss about termination of DNA replication.